Okay, first of all, I'd like to thank the organizer for inviting me on behalf of Ricardo to the annual Congress of the British Society for Allergy and Clinical Immunology. Secondly, I send a big hug to Ricardo. In this slide, you can see my conflict of interest. I am employed at the Istituto Dermopatico dell'Immacolata in Rome. I received a research grant from Italian Ministry of Health, financial support for consultation from Stallergen and Thermo Fisher, and non-financial support from the macroarray diagnostics. This is the list of topics I will cover in my report, mainly concerning some controversial aspects of the lipid transfer protein allergy. But first of all, I would like to underline some historical hints regarding the main aspect of the lipid transfer protein allergy. We know that lipid transfer protein family of allergenic proteins represent an important airborne and food allergen, particularly found and spread in the Mediterranean area. Such molecules are highly conserved molecules involved in plant defense against fungi and bacteria. They are stable, compact, because of the presence of 8 conserved cysteines that confers an extreme resistance to gastrointestinal proteolysis and thermal processing. And such physicochemical characteristics are linked with their ability to determine and increase the risk to develop systemic and severe reaction to food, even after physical exercise. There are 2 distinct subfamilies of lipid transfer protein currently known, discriminated on the basis of the molecular weight. But the majority of non-allergenic proteins belong to the first subfamily, having the higher molecular weight, 9 kilodalton, 47 out of the 51 lipid transfer protein currently recorded in the IUIS database. Since such molecules are homologous molecules, there is a various degree of cross-reactivity, mainly on the basis of the botanical relationship among these molecules, with the total exception of the lipid transfer protein belonging to the literature of the wall, because such molecule has no identity, no similarity compared to the other lipid transfer proteins. We started in studying such molecules extensively by means of macroarray system since 2007, when there was only one lipid transfer protein spotted on the array, group 3 from pitch, we could immediately notice how this molecules was the top ranked food allergen found in our patients, the major responsible for adverse reaction to food, accounting for about 10% of the total allergic population. We also verify and observed that in the following years, with the increasing of the molecules available for diagnosis in the chip, also increased accordingly the number of patients properly detected and evaluated by such methods. And this is because lipid transfer protein are homologous molecules, but not identical molecules. And the degree of cross-reactivity follows the rule of the botanical relationship. In fact, if you compare group 3 from peach with the other Bsaci, for example, lipid transfer protein from plum, cherry, or apricot, you can find very high levels of identity. On the other hand, if you try to compare the structure, the amino acid structure of PP3 from peach with biological sources that are far away from a botanical point of view, for example, the citrus fruit, you can find no more than 10-15% of identity. And in fact, in the real life, when you encounter a patient having lipid transfer protein reactivity, quite rarely he refers for problem for adverse reaction to orange, for example. And when you have a patient having at the same time problems with peach and orange, you have to think about to maclin and not lipid transfer protein. But this is total another story. This is the list of lipid transfer protein currently registered in the IUIS database. As I have already told you, a number of them can be evaluated by means of macroarray system. But quite recently, another macroarray system has been introduced in the market, containing even more molecules to be tested. And therefore, we evaluated in 140 patients in duplicate using the same biological sample, the two methods. And we could observe some interesting things. For example, first of all, only in 62% of cases, the two methods could detect at the same time patients. And it is understandable, conceivable that 23% more patients can be observed and detected with the array containing higher number of lipid transfer protein available for diagnosis. It's quite surprisingly to me observed that 15% of patients are detected only with the first method that contains lower number of lipid transfer protein, and all the lipid transfer protein represented are present at the same time in the second test. There are several possible explanations for this. First of all, I have no data, no information regarding the kind of isoform used and chosen for the diagnosis in these two systems. Secondly, the spotting strategy is totally different in the two systems. For example, in the first system, the molecules are spotted in triplicate directly on the slide. In the second system, the molecules are firstly coupled on nanobits, and then the nanobits are placed on nitrocellulose membrane. And third, but not last, also the detection system is totally different. Fluorescence in one method, in the older method, and enzyme linked alkaline phosphatase driven in the other methods. And therefore, all those variables could affect, could influence the outcome of these methods. And this should be considered always when you manage patients in the daily clinical practice. Talking about the clinics, I'm going to share with you several data, retrospective data, a retrospective overview of more than 6,000 patients evaluated in our institute by means of microarray system, by means of the radar chart, which is a graphical approach that you can use to visualize at the same time more observation with a two-dimensional chart. And every single spoke that you observe here represents one of the molecules studied. And the length of the spoke is proportional to the respective odds ratio of the variable relative to the association with the clinical symptoms that you are observing, that you are checking. And for example, if we focus our attention on patients having severe reaction to food, as you can see, lipid transfer protein are always directly related with the presence of lipid transfer of severe reaction to food. And on the other hand, profilin and PR10 molecules show value below 1, and therefore they are not directly related with the occurrence of severe reaction to food. On the other hand, when you encounter patients having oral allergy syndrome, in this case, as you can see, profilin, PR10, lipid transfer protein are all directly related with the occurrence of local symptoms to food. And thus confirming the possible protective role of profilin and PR10 for the aggressivity, for the danger represented by lipid transfer protein sensitization. But such situation could be totally reverted in the case of several cofactors, such as, for example, physical exercise within 3 hours the ingestion of food. And what about the geographical differences? Okay, in the late 1990s, we believed that lipid transfer protein reactivity was just a Mediterranean affair. But when we compared in collaboration with Isabel and Stephen Till, our patients, our reactive patients to lipid transfer protein, the IG sensitization profiles of our patients with those observed in the United Kingdom, as you can see, the IgE sensitization profile was absolutely the same. And PUP3 from pitch being the molecule most frequently observed found positive in patients belonging to both countries. But if the main source of lipid transfer protein reactivity and probably sensitization is the peach in Italy, this is not true for United Kingdom, where tomato, tree nuts, apple, and seeds and so on and so far are associated with the presence of lipid transfer protein reactivity. And this observation teach us that not always when you find the molecules that are most frequently found positive, most frequently reactive, this not always means that that given molecule is the primary sensitizer. Just means that that molecule seems to work better in that system than if compared to other molecules. What is certain for sure is that in Northern Europe, LTP sensitization is rarely associated with severe reaction to food, probably because of the high prevalence of PR10 sensitization. And it's also interesting what happened in northern China, where the lipid transfer protein reactivity is always associated with mugwort allergy, which is widespread in the tree. In fact, in this wonderful paper, it has been demonstrated that R23 could totally inhibit the signal linked to PUP3 sensitization. And therefore, it has been suggested that the primary sensitization for LTP allergy in northern China is driven by the pollen, mugwort pollen. And this model, this paper, this study led me to move on to the next topic, the controversial question of the primary sensitization to LTPs. Quite often in the past, we observed that when you have at the same time patients that are reactive to mugwort, plant tree, and olive tree, you are in the presence of a lipid transfer protein sensitization. And therefore, we studied more carefully such patients. And starting with olive tree sensitization, we studied a cohort of 839 patients extensively by means of molecules. And the major allergen of olive tree, olive 1, reactivity, could identify the specific olive tree pollen allergic population with the respiratory symptoms, as you can see here, but that such reactivity was never associated with the presence of food allergy. And on the other hand, when you observe, when you study patients reactive to the lipid transfer protein from olive pollen, olive 7, in this case, patients were associated with the local or systemic reaction to food and never with respiratory symptoms. So two distinct, two opposite clinical behavior, and it's quite difficult to me to believe that one pollen could sensitize two different clinical subsets. And in the case of plant tree, we could observe absolutely the same picture, the same scenario. The major allergen PI2 associated with respiratory symptoms and never with food allergy. On the other hand, the lipid transfer protein from plant tree, PI3, associated with local and systemic reaction to food. Moreover, we conducted a retrospective study along Italy involving 25 centers and more than 8,000 patients together with Riccardo. And we could observe that among PUP3 reactors, patients reacted to the lipid transfer protein from plant tree and mugwort always largely exceeded that of patients sensitized to the respective major pollen allergens, for example, at R21 from mugwort and PLI2, PLI1 from plant tree. Moreover, PUP3 reactivity remains stable, as you can see here, around 74 to 90% at all ages. And mugwort and olive tree lipid transfer protein reactivity was almost missing in younger patients. And plant tree sensitization was always far below the levels observed for PUP3. And PUP3 Ig levels always exceeded Ig pollen LTP values at all ages. Moreover, when we try to replicate the experiment made in China, trying to inhibit the signal of PUP3 with the mugwort, commercial extract from mugwort and plant tree, we failed. And the PUP3 Ig levels always exceeded RP3 Ig levels in those patients that had both reactivities in 84% of cases. And all patients positive to only one lipid transfer protein at the baseline since childhood were sensitized to pupil-3. And all those observations are convincing points that support the idea that lipid transfer protein could act as a primary sensitizer, at least in our region. But it's possible that in other regions with other not endemic, where there is not endemic LTP sensitization, where other population, other haplotypes are present and other kind of exposure to allergen, LTP reactivity might be initiated by inhalation or contact of hemp LTP, the CAN S3, even via passive smoke, as demonstrated quite recently in adolescent and pediatric patients. And final point, final topic, the hierarchy in LTP sensitization. Okay, in 2015, we started together with Stefan Thiel a large number of patients having lipid transfer protein reactivity, and we could observe a wide heterogeneity in molecular recognition in these patients, from monoreactivity to multiple LTP recognition. In the case of monoreactivity, in the vast majority of cases, as you can see here, the reactivity was directed against the pupil-3 from pitch. And another important observation was the fact that when we had patients having multiple reactivity to more than 5 molecules, such condition was significantly associated with systemic or local reaction to food. And therefore, LTP hypersensitive patients could show a spectrum of co-recognition from monoreactivity to multiple reactivity. And this process seems to follow a precise and predictable route, a predictable sequence, starting in the vast majority of cases since the childhood with peach, and then following with other Bsaci fruit, and then tree nuts, seeds, and peanuts, afterwards cereals, and finally vegetables. And these are the most dangerous patients capable to recognize more molecules and therefore more fruit, more food, even unrelated, even botanically unrelated with peach. In conclusion, historical data testify for high frequency of Bsaci allergy, high prevalence of systemic symptoms when you have to manage with patients having lipid transfer protein reactivity. Such picture was partially modified with a larger study and larger observation, including other countries where lipid transfer protein reactivity was found. And in that country, a huge variability in clinical expression was found, from a complete absence, clinically irrelevance of lipid transfer protein reactivity, to very severe symptoms. And this could be, this observation, this clinical outcome can be, for some instance, affected by protective effect exerted by co-sensitization to other panallergen vegetables, and on the other side, cofactor that could enhance the risk to have systemic reaction to food. And in this regard, I can try to depict a sort of hopefully useful algorithm for the daily clinical practice. So when you encounter patients having wide ability to recognize in recognizing lipid transfer proteins and monoreactivity to lipid transfer protein without any other vegetable panallergen sensitization, okay, you are in the presence of patients having high risk to develop systemic reaction to food. And on the other hand, the opposite. When you have patients having scant capability to recognize lipid transfer proteins and the sensitization to profilin, to PR10, you are in the presence of low risk to develop systemic reaction to food. But you have to alert your patients to avoid possible association within 3 hours the ingestion of the culprit food with physical exercise, or, for example, anti-inflammatory drug ingestion. There are geographical differences. There are regions that are considered as endemic for LTP sensitization, and allergy prevail in Southern European countries. And the relevance of such reactivity is associated also with the controversial question of the primary sensitization, because we have convincing data supporting the role of PUP3 as a primary sensitizer for lipid transfer protein. There are convincing data for R23 as a primary sensitizer in northern China. Other supporting information regarding the role of cannabis, can S3 lipid transfer protein in induction of LTP sensitization in Northern Europe, in Belgium, for example. Therefore, possibly you can find several different situations in several different conditions. And last, the hierarchy in LTP sensitization, we quite often observe a sort of spectrum of co-recognition that quite often starts with the pitch reactivity, which is followed first of all by botanically related fluids, and afterwards by botanically non-related fluid in multiple sensitizers. And with this slide, I'd like to thank you very much for your so kind attention.